The overall goal of this procedure is to describe two in vitro developmental toxicity test systems based on human embryonic stem cells and transcriptome studies. This is accomplished by first culturing human embryonic stem cells on mouse embryonic fibroblasts. The second step is to either let the human embryonic stem cells form embryoid bodies in which they can generate cells of all three germ layers, or differentiate them into neuroectodermal progenitors. Next, the embryoid bodies, or neuroectodermal progenitors, are treated with compounds of interest at different concentrations during differentiation. The final steps are to assess the viability of the cells and isolate the RNA. Ultimately, transcriptome microarray analysis is used to show the changes in RNA expression of cells treated with a compound of interest compared to untreated control cells.
Based on human embryonic stem cells and DNA microarrays, we have established an in vitro methodology allowing monitoring of human-relevant developmental toxicity effects. The methodology can be applied by the pharmaceutical and chemical industry for safety screening of potential drugs and environmental toxicants.
This method can help to answer key questions in the field of developmental toxicity, such as the effect of chemicals on the very early neurodevelopment.